Next is structure of protein. Proteins are already you read, that is the polymers of the amino acid, that is alpha amino acid, polymers of the alpha amino acid. They are connected with the peptide bond or the peptide linkage. See, they formed between the COOH group and the NH2 group. The reaction between the two molecules, the similar or different amino acids, proceed through the combination of the amino group. That one molecule will be the carboxyl of each other. The resulting the elimination molecule formation of the CONH bond. That is, the product of the reaction is called dipeptide bond, because for example, glycine react with alanine. Glycine react with alanine. I will write the NH2 like this. Glycine and alanine. See, here the removal of water molecule from these two compounds, you will be get the CONH group. The CONH. This is the peptide linkage. So loss of the water molecule, and you will get glycine alanine product. So this linkage is called peptide linkage. Peptide linkage, and the name of the compound is glycine alanine. Glycine alanine or glyala. The three-letter symbol is glyala. This forms a dipeptide. This is a dipeptide because of 2 carboxylic acid. In case of 3 carboxylic acid present, means tripeptide. 4 carboxylic acid, tetrapeptide. 5 pentapeptide. Like this, 10, more than 10 means simply we can tell the polypeptides linkage. So this is the structure of protein. The structure of protein mainly classified on the basis of the molecular shape. First, the fibrous protein. Fibrous protein and the globular protein. Mainly the two types. The first fibrous protein. The polypeptide chain run parallel and they're held together by the hydrogen and disulfide bond. Hydrogen and disulfide bonds. Then the fiber-like structure is formed. Usually the fiber-like structure is formed. Such proteins are generally insoluble in water. These are insoluble in water. Insoluble in water, and so common examples of keratin, that is where it is present mean in hair, wool, silks. Then myosin is present in muscles. So these are called the fibrous protein. Then the globular protein. The globular proteins, when the chains of polypeptides coils around to give a spherical shape, like a DNA structure, like a DNA structure. See, these are usually soluble in water. It's opposite. This is soluble in water. Soluble in water, but examples insulin and albumins. Insulin and albumins. The common example for the globular protein. See, the structure of say protein can be studied the different ways, like the secondary primary structure, secondary structure, tertiary structure, and quaternary. So totally the four different levels are there. First we will discuss the primary structure. Usually the primary structure of proteins having one or more polypeptide chains. They have one or more polypeptide chains. That means that each polypeptide in a protein has an amino acid linked with each other. Already we linked. That means the specific sequence and the sequence is amino acid that is said to be the primary structure. That sequence are created different proteins. For example, we can see that in our textbook, in NCRT textbooks are given here. The structure like this. The NCR, you can look at the NCRT textbook just like this having the peptide linkage. So this sequence of amino acids are here create a different protein. Then usually in your textbook, NCRT textbooks are given the primary structure like this. So these are the primary structure of the protein. Then the secondary structure. The secondary structure proteins refer to a shape which is long peptide chain, that is the polypeptide chain can exist. That is they are found in exist in the two different structure like alpha helix and beta pilated structure. Two different alpha helix and beta pilated structure. These structures arise due to regular folding of the backbone of the polypeptide like this. The polypeptide chain, that is here the hydrogen bond between, look, the hydrogen bond between here, the hydrogen bond between the C double bond O, this C double bond O and the NH group, the hydrogen bond between this. So this peptide bond, this type is called alpha helix structure. You can see in your textbook. The polypeptide chain forms all possible hydrogen bond by twisting into right-handed screw, that is screw like this, twisting into right-handed screw with the NH group, this is like NH group, with the NH group of amino acid residue hydrogen bonded to the C double bond O, that is the adjacent turn of the helix. Okay, here the in beta structure, in beta structure all peptide chains are stretched out to nearly the maximum extension and the side by side, which are held together by the intermolecular hydrogen bonds. So the structure resembles the plated folds on the that is the beta pilated sheet. You can see that in your NCRT textbook the structures are given. Then tertiary structure. Third one is tertiary. Primary and the secondary structure is this. This is secondary and this is tertiary structure. This tertiary structure of proteins. See, this structures represent the overall folding of the polypeptide chain. The overall folding of the polypeptide chain means like See this, the primary structure like this, the secondary structure like this, and the tertiary structure, the overall folding like this. So this is the tertiary structure of protein. So here that is the folding of the secondary structure, that is further folding of the secondary structure, just folding to the secondary structure, look like here, just look like. Okay, so this is a further folding of the secondary structure is called, that is tertiary structure. It gives rise to two major shapes, that is fibrous and globrous. The main force which is stabilized the secondary and tertiary structures of proteins are hydrogen bonds and the disulfide linkage. There is the van der Waals forces and the electrostatic forces of attraction is also there. Then the quaternary structure. The fourth one is quaternary structure of protein. See, some of the proteins are composed of the two or more polypeptide chains referred to as subunits. The spatial arrangements of these subunits with respect to each other. So these are the quaternary structure. The quaternary structure, that is the like this. Just a joining of this and one like this. So this is the different structure: primary structure of protein, secondary structure of protein, tertiary structure of protein, and quaternary structure of the protein. Then the denaturation of protein. The protein found in the biological system with a unique three-dimensional structure, that is, and the biological activities are called native protein. Okay, these are the native protein. When the protein is its a native form, it's subject to a physical change, like a temperature or chemical change, like a pH value, hydrogen bonds, or disturbed means. That is due to the cell, the unfold helix and uncoiled proteins loses the biological activity. So these types are called denaturations of the protein. Then during the denaturation, secondary and the tertiary structures are destroyed. Secondary and tertiary structure are destroyed, but the primary structures remains intact, does not affect. So these are called denaturations of the protein. Then the common example, coagulation of the egg. The egg white on boiling, a common it's become a solid, liquid become a solid. That is curdling of the milk, which is caused due to the formation of the lactic acid by the bacteria present in the milk. So these are the common examples of the denaturation of protein.